Hello, welcome back. Right, so we're going to focus on another biological molecule called a nucleotide. And nucleotides are the building blocks of DNA. So DNA is a polymer, or we can call it a polynucleotide. It's made from many monomers, and a nucleotide is a monomer. So lots of monomers put together make a polymer. So another example you've looked at before is amino acids are monomers. If you stick lots of amino acids together, you get a polymer, which is a polynucleotide. A polypeptide. Okay, so quick intro to DNA. You probably know a little bit about it already. It's found in the nucleus of our cells, and it carries really important genetic information. One of them, obviously, is for inheritance, passing down genetic information from one generation to the next. And another important aspect of what this molecule does, it's storing really important genetic information. It's carrying the instructions on how to make different proteins. Protein synthesis cannot happen without the genetic information found inside this DNA. So we're going to look at just the building blocks that make it up and how they bond together. So a nucleotide is made up of three parts. So it's got a pentose sugar. So you've looked at hexose sugars already. We looked at glucose. This is hexose with 6 carbons. So pentose, it's only 5 carbons. I should say 4. There's our 4. And then carbon 5 is up here. So that's the first part. It's the pentose sugar. And then it's got a phosphate group connected to carbon 5. There's our phosphate. And then finally, it's got a nitrogenous base. And there's only 4 bases. When we talk about DNA, there's 4 we need to know. A, T, C, and G, or adenine, thymine, cytosine, and guanine. All right. So let's look at how these monomers or these mononucleotides connect together. So looking here now on this image of the DNA here, you can see it's twisting around itself. We call this, it's a helical shape, and it's a double helix because if you look, there's 1 strand and there's 2 strands. So I'm not going to show any twisting happening. All I'm going to show you is how an individual strand forms first. So if we're talking about how the strands build, well, the mononucleotides, they're going to form condensation reactions, which is when the molecule of water is lost, and it's going to form something called a phosphodiester bond. So let's do a phosphodiester bond now. So there's carbon 3. So carbon 3 is involved, and carbon 5 is involved as well. All right, so there's our 1, 2, 3, 4, and 5. So you can see here, carbon 3 and carbon 5 are bonds together. And you're going to do a much better job than I am at drawing these. That's wrong. There's my pentose sugar again. And there's our nitrogenous bases. So that could be A, that one. That could be C. Doesn't really matter. I can make that C as well. So now that's how we draw a single strand so far. So the single strand has been formed, and we call this a backbone. It's where carbon 3 and carbon 5 are bonding together, and we get something called a phosphodiester bond. So there's, see there, that there is a phosphodiester bond. All right. If you want to break that bond, you'll have to add water, and we call that hydrolysis. So it's, all of these molecules so far I've looked at, there's always been condensation and hydrolysis in terms of forming and breaking. Right, so the 4 bases are adenine, thymine, cytosine, and guanine. We could just use these letters A, T, C, G if you want. A little bit you need to know about these bases. They come under as 2 different types. We've got one called purines and one called pyrimidines. So I'm going to talk you through the pyrimidines. The best way to remember it, it's a bit of a strange way to do it, but it works for me. I want you to think for the pyrimidines, think of a pyramid, and it's a really sharp top. If you were to stick your finger on the top of this really sharp pyramid, there's a good chance that you would cut your finger. So that's a way to remember that the pyrimidines are cytosine and thymine. And these, the pyrimidines, they are the small nucleotides. So they've only got one ring structure, you can see here. You don't need to be able to draw this. Just being aware that we just draw it as these little rectangles, that's fine for now. But just being aware that pyrimidines are the small, they're the small nucleotides because they've only got a one ring structure. Whereas the purines, well, what's left? If there's C and T are the pyrimidines, we're left with A and G. They are the purines, and they are bigger because they've got a 2-ring structure. All right, now we're going to try and draw a double helix. So we'll just get rid of this. So when drawing a double helix, that means now we have to draw 2 strands, and the way those 2 strands form, it's something called anti-parallel. So while one strand is going up, so think of this pentose sugar, this top bit is like an arrow pointing up. So that's pointing up, just like last one we just did. Pointing up as well. And there's our bases. Okay. So the other strand is going to form bonds. So the nitrogenous bases that are complementary to the ones on the left are going to form hydrogen bonds. So the complementary bases are, if ever you get adenine or A, the only other base that can bond with it is T. And if it was G, only C can bond with it. So if it was the other way around, if this was C, then this would have to be G. All right, that's the rule. So A always goes with T, and C always goes with G. Now, this is the difficult bit. We have to draw it slightly differently. We can't draw the sugars pointing upwards. They're now pointing down. So let's do down there. So this is worth practicing doing. It's quite tricky at first. So they're pointing down. So now we can draw our connected ends together. Now we're forming our phosphodiester bonds. And there is the start of you drawing a double helix, or not the helical part, but just the double strands. We've got our phosphodiester bonds here, forming the backbone of each strand. And we've also got the hydrogen bonds between the actual bases. So, key thing here, know the different bases. There's 4 of them. Know the 3 different parts that make up a nucleotide, the pentose sugar, the phosphate and nitrogenous base. Knowing the 2 types of nitrogenous bases, the purines and the pyrimidines, and what the differences are. I use this pyramid and how you cut yourself, if it was sharp, to remind yourself which 2 bases are the pyrimidines. And then learn how to draw this anti-parallel structure, and anti-parallel being while one strand is going up, the other one is going down. All right, good luck with that.